Hi, welcome back to Bio 131. Today we're doing the first of three parts that I've taken Chapter 27 on prokaryote diversity and divided it up into three separate lectures. And the first one is going to be basic introduction to cell theory, one of the basic ideas in biology, and then the structure of what makes a prokaryote cell a prokaryote cell. If you've had 130, this will probably be a bit of a review for you. The second part is going to be on motility, how prokaryotes move around, and on DNA and replication. And then the third part is taken up of not in depth, but a general introduction to various types of metabolism that prokaryotes have. And so let's get going and talk about some prokaryotes. And so the two groups that are prokaryotic, and remember in lab we went over the three domains of life, and eukaryote cells and eukaryote organisms make up one. The other two domains have what we call prokaryote cell structure, and they are the archaea and the bacteria. And so one thing to first start thinking about is that they are everywhere. There's no habitat that has any life on it that archaea or bacteria are not growing there, and they can be some of the most numerous organisms on Earth, and they're found in all sorts of habitats. Okay? So here I want to start with, and these are two cells that we saw in our first lab with microscopes and looking at how to make slides. And this is Elodea with little chloroplasts going around, and then a stain of human, my cheek cells here in the corner. And one of the first theories we want to talk about, and remembering from last time, a theory is a basic explanation that has been supported by evidence. And one of the basic theories in biology is the theory of cells. Basically, it's a very simple theory in that all life is made up of cells. And when we get to our third part of this, we'll be talking about viruses and viroids, and then technically we have to talk about why they aren't, quote, alive, because they really aren't cells by themselves. So all living things are made up of cells. Some organisms, now both of these examples I have on top of the slide here are of multicellular organisms that we had in lab that day. But when we're talking about prokaryotes, we're talking primarily, if not exclusively, about single-cell organisms. And there are lots of eukaryotic single-cell organisms as well. So all organisms are made up of cells. Some, the whole organism is a single cell. Many larger, all the larger living organisms we see are made up of smaller cells combined into that one large organism. Right? Second part is the explanation. One explains the structure of life is made up of cells. The second explains how life continues and reproduces. And that is quite simply that cells come from other cells, right? So if we're looking up at these plant cells, they were made by other plant cells. If you're looking at these cheek cells, epithelial cells, they were made by other ones down at the base layers below. And as the ones slough off, new ones are forming down at the base. Even when we get to reproduction, and we'll be talking about different types of sexual and asexual reproduction, usually gametes are involved in sexual reproduction. Those are single cells that combine into a new cell, a new genetic organism, but cells are always coming from cells. And so the way cells reproduce is in three basic mechanisms that we'll talk about. And the first applies to what we're talking about today, and that will be for prokaryotic cells. They divide by a process called binary fission. And I'll draw that out and talk about it when we're going over DNA in the second part of this chapter's lectures. And then we'll learn about two different ways that eukaryote cells divide. All of the growth and asexual reproduction is by mitosis, a cell making an identical copy of itself, right? And then in meiosis, we'll have to stop talking about some terms like diploid and haploid and sexual reproduction. Those two terms, so meiosis is involved in sexual reproduction. Those two terms only apply to eukaryote cells. All right? So one very basic thing to remember, prokaryote cells, all bacteria, and archaea divide by binary fission. Right? Eukaryote cells that have nuclei in them, when we get to it, those divide by either mitosis or meiosis. And so we have two very different types of cells, and I've been using the terms already, prokaryote and eukaryote. Karyo means seed or kernel, and so that refers to the nucleus. Pro means before, right? And so a prokaryotic cell, the term is implying that they came about before nuclei, and therefore they don't have a nucleus. Eukaryote, the EU means true, In this case, that would be a true nucleus, meaning they have a nucleus. And those would be eukaryote cells, right? And so features of prokaryote cell, they don't have a nucleus, but what we'll describe that as is they lack membrane-bound organelles. So they don't, not only do they not have a nucleus, they don't have a chloroplast, they don't have mitochondria, a whole bunch of things I'll list in a minute. Eukaryote cells, the way we recognize are all these things I was listing out. They have a nucleus that contains the DNA, and what is contained in is a bubble, right, of membrane. And they have several other organelles, each of which is contained and separated from other parts of the cell, each with its own function, surrounded by membrane. And so, and cut off completely by itself, right? And those are membrane-bound organelles, and that's what makes them a eukaryote. And the feature I have down here is that prokaryote cells exist usually as single cells or small little chains coming together. So prokaryote cells are single cells or small chains. Eukaryote cells can be single cells, and we'll see a lot of that in diversity, but everything that you see that's multicellular, easy to see with the naked eye, that's all eukaryotes. And the big multicellular kingdoms are plants, animals, and fungi, right? So those are eukaryotic. Okay, prokaryotic, simpler, smaller, simpler structure. Well, I'm going to draw one in a minute, right? Smaller cells. And by simpler structure, I mean they don't have all of these internal membrane-bound organelles, right? Eukaryotic cells, lots of membrane-bound organelles. And so we're going to learn two types of cells that are prokaryotic, and they're whole domains. And so even though I'm just drawing one word for bacteria here, there's more diversity in the bacteria in terms of how they do their metabolism and exist in habitats in life than exists in all of the eukaryotes, right? And if we were taking a microbiology class, I would list out all sorts of different types of bacteria. But we're just going to keep it at the highest level of domain and just talk about bacteria, right? At the structural level, the way I'm describing it, you cannot tell a bacteria from an archaea, right? Because they're both prokaryotic in terms of their structure, right? I will give you features that they have, some features about the differences in their cell wall, things like that, that will list the difference between them. But just structurally, if I were to draw them both out for us and for what we're looking at, you couldn't tell the difference between an archaea and a bacteria, right? So prokaryote. For eukaryotes, they include a whole diversity. So of course I mentioned the animals, the plants, and the fungi as the multicellular kingdoms. The other ones that are pretty big, so you could go down to the seashore on our low tide, you see red and brown algae, so the kelps and the seaweeds, those are quite large, right? And so they're also eukaryotic. And then in our eukaryote diversity lab, we're going to go over lots of single-celled organisms, diatoms, amoebas, euglenas, paramecia, animal parasites like malaria, right? Oomycetes, water moles, and there's just a huge diversity of dinoflagellates that will be seen that are all single-celled organisms. So the eukaryotes, whether you're single-cell or multicellular, isn't the defining feature of the domains, it's the structure of the cells, right? Because there's lots of single cells that are eukaryotes, right? If it is multicellular, it is eukaryote, not prokaryote. But just because that it's a single cell, you can't say that it's eukaryote versus prokaryote. Okay, so their structure is less complex. Let's take a look and see what that looks like. And so I'm going to start with, if I can find my pen here. I'm going to start with drawing one of the features, and I'll draw it in black, and this will be, oh, I guess I'm starting with yellow, the cell wall. Right? And we'll compare the cell wall of eukaryotes and prokaryotes. And within the prokaryotes, there are two types, and I'll do bacteria, and the cell wall in the bacteria is made up of a substance peptido. And so what peptidoglycan is, it's made up of simple sugars put together, but then it also has amino acids joined together. So it has polypeptide. That's more 130 stuff. So you just want to remember the term peptidoglycan. right? That's in the bacteria. The arc in the eukaryotes do not have that. The next feature I want to go over is inside the cell wall, internally, will be the cell membrane. And you learn lots about the cell membrane in Bio 130 its cell structure. But for us, what's important about the cell membrane, first of all, it's made up of phospholipids. So it has a lipid part that repels water and a phosphate part that likes water. And those properties give the membrane its structure. But what it's really doing is it controls what gets in and out of the cell. So far, this isn't enough to tell the difference between a prokaryote and a eukaryote, because many eukaryotes also have a cell wall like a plant, and inside it they would have a cell membrane. Right? Next thing I'm going to draw is that it has a single, I'll keep it very simple, single loop DNA. It has one single chromosome. That's different when we get to eukaryotes. Eukaryotes, like you, have multiple chromosomes, right? And also when it goes to divide, the chromosome will attach onto that cell membrane. The other structure it has is, in order for the DNA to be expressed, there's an organelle called a ribosome. And what a ribosome is, it takes that DNA information, right, and converts it into a protein. And that, how it does that is in a process called translation and transcription. Transcription to make RNA and translation to make the protein. That's covered in Bio 130, right? But we just want to remember that they have ribosomes, and the ribosomes in a eukaryote and the ribosomes in a prokaryote do the same thing. However, if you look at the structures of them, they're slightly different. And you can tell the ribosomes coming from a bacteria because they're smaller in size than ribosomes coming from a eukaryote. And that'll be important when we get to talk about how we got some of the organelles in the eukaryotes later on. Okay, and then I'm also going to add here is that the bacteria have flagella, not all of them, but some can. And these flagella, when we get to them, are rigid, they're very strong, right? But they're not flexible, and they actually move like rowing a boat with oars. They will spin at the base. Okay, so let's go back and take a look at some prettier pictures of what these look like. And so you can see here in the inside region where I drew the DNA, that would be called the nucleoid, right? So O-I-D at the end, nucleoid, not nucleus. And if I go back to my diagram, you'll notice there are no membranes. So if I go back, so if red was my membrane, notice except for the outer cell membrane, nothing on the inside has membranes around it. So the DNA is not surrounded by red. In a eukaryote cell, it would be, and that would be the nuclear envelope. Right, so we have the cell wall to the outside, then you have the flagella. Other things that they have, and that we'll go over, because you notice to the outside they have some extensions called fimbriae, right? They're showing you the little dots that are the ribosomes. Then it's also labeled there a capsule, and that's a protein, kind of viscous, thick protein layer to the outside. Okay, now what we're looking at is a eukaryote cell, and this is what I was talking about earlier when I said membrane-bound organelles. So here, all of these lines that are in purple, blue, green, and orange, those are all membrane, right? Surrounded by the cell membrane of the outside. So there's lots of structures, right? So that's where we talk about the structure of a prokaryote cell being simpler, right? My diagram didn't include nearly all of that detail that we have in a eukaryote cell. And this is an animal-type eukaryote cell. Here's a plant-type eukaryote cell showing it as a cell wall, right? And other additional features, right? So this is from another chapter. This is just showing you that eukaryote cells at the cell level are much more complex, right, in terms of cell structure than the prokaryote. Okay, so prokaryotes, nearly all of them have a cell wall. And the way they grow in the environment, they can exist in more environments than any other group of organisms, meaning the eukaryotes. And so we find them in all sorts of different areas, some rather extreme, like even very deep in the soil, frozen up north, all sorts of areas, right? Boiling hot springs. But when they usually grow, they don't grow as single cultures. They grow in combination with other, in this case, bacteria, and they form what we call biofilms. And basically, there may be a bacteria that's doing photosynthesis and releasing some products that others are using as energy to feed, so they feed on the photosynthetic bacteria. So if you imagine a field, and you have the plants in the field, and the mice eating the plants, and the snake eating the mice all coming together, if you took that interaction and reduced it down to the thinnest film of these different layers, and these cells, so you have some photosynthetic, like the grass in the field, you have some photosynthetic type bacteria. Then you have the cows eating the grass, the mice before, the mice eating the grass. There'd be another one eating the photosynthetic bacteria. So species A would be photosynthetic bacteria, species B would be eating them, right? And then species C would be eating them, right? And so they form this complex interaction, which we call biofilms, right? Again, by definition, they're typically smaller than eukaryotes, and they lack these internal membrane-bound organelles. And as I mentioned, the ribosomes are smaller in size. Right now, this is a tidbit information, but that fact will be important later. All right? They're tremendously important on Earth. If we got rid of all of the prokaryotes, right, life would stop. Right? When we get to biogeochemical cycles, like the one for nitrogen, so we like to think of plants being the base of the food chain, right? And plants move up energy, but plants are also the source of protein. All right, and so the cow gets its energy from eating the grass, but it's also getting all of its protein by eating the grass, and the grass makes the protein. And in order to make the protein, it needs nitrogen, and the only way it can get nitrogen is from bacteria in the soil. Right? So they have tremendous impact on the earth, right? Overall, very few cause diseases. That being said, there are a lot of bacterial disease, but it's a fraction of the overall bacteria that are there. But there's some very, very serious bacterial diseases. I already talked a little bit about the biogeochemical cycles. We'll go over that more in the third lecture on this chapter when I'm talking about metabolism, and that's an example of nitrogen metabolism. Right? They form very important relationships with other prokaryote organisms, as I mentioned in before with the biofilms. But also, when we get to these biogeochemical cycles, they form associations with eukaryote organisms, such as nitrogen-fixing bacteria that are in plant roots that help them obtain this nitrogen that I mentioned before to make their proteins. And one of the evidence that mitochondria and chloroplasts, which are organelles in eukaryotes that we'll cover in the next chapter, right, that they came from bacteria is the fact that they have these small ribosomes. So we'll be talking about that. But that may be an example of an extreme close relationship between the two of them where they've actually become incorporated inside and become part of the cell. But we'll get to that next chapter. Okay, here's some examples of biofilms. I think I have one more there. They become a big problem up in the space station. And so they're actually doing all sorts of investigations because biofilms, like plaque around your teeth, they can make it very hard for just use medicines and things to fight disease-causing organisms and make it very resistant to find places for these disease bacteria to hide. They also can break down equipment. So in the space station, it's more the breakdown of equipment because these bacteria are degrading things that are up there. Here's a marble monument, and you can see this film on the outside. And what it does is they start breaking down the actual marble, somewhat soft rock, and again, they're very hard to treat. They also can form films that in the presence of one that by itself, that could be a disease-causing organism, that presence of that one bacteria by itself might be easy to treat. The presence of the other ones break down the antibiotic so that you can't kill the disease-causing bacteria. And here's one that's causing infection in skin, urinary tract, and lungs. And when it's present as a biofilm, it's much more difficult to treat. Okay, origins. Prokaryote means before nucleus. And when we look at a big picture of life, the prokaryotes came before the eukaryotes. And so prokaryotes, the first living organisms, probably go back about 3.5 billion years. Eukaryotes only go back a little over 2 billion years. So for all of life on Earth, just about half of it was all prokaryotic at the time. All right? They were the earliest and simplest of the living things. I just want to point out, when we get to evolution, everything that we need for life was figured out by prokaryotes. And so how we get a prokaryote from nothing is a huge question. But how a simple prokaryote gets more complex and turns into a eukaryote, we have some evidence of that. And then how these multicellular organisms form from that is much easier to explain than how you get a prokaryote from nothing. Right? So the simplest living organisms that figured out all the basics of life, how DNA is going to be used, the cell wall, these things I mentioned before, the ribosomes, all those others. Right? So that all came with prokaryotes. They're in all habitats, different types of metabolism and photosynthesis, which we'll get to in our third part of the lecture. And so in lab, we're not, so if you take a class all by itself down the hall, if we're in the building, it would be microbiology. So they're important enough, especially in medicine, that, or medical fields, allied health fields, that you take a whole separate course just on them and their diversity and how to identify them and their types of metabolism. So we're just going to touch on the highest level of it. They have three basic shapes. They're either kind of a rod shaped, it's more of a brick shaped. They're round, which is coccus, or they can be helical or spiral as single cells. Generally, the single cells, sometimes they can stick together in little clusters, and we'll go over those terms in lab. So you can get, if this rod-shaped would be bacillus, if two of them stick together, it would be diplobacillus. If they go into a chain, all right, it'd be strepto. If they go into little patches, it'd be staphylo. So sometimes they stick together. They can stick end to end to make what we call filaments. So they form small little chains of cells, but that's about as complex or as multicellular as they get. And so here are three different shapes. Electron micrographs colored shows you three different shapes. And again, we'll be going over this in lab, and you'll see them under the microscope and see the three different shapes. We're going to take advantage of one feature of the cell wall when we get to it and do a gram stain. Okay, so this is again going over to the outside. We have some extension. So the cell wall here is an orange, right? And this is a bacteria. And so that means that cell wall is made up of, remember the term, peptidoglycan, right? And we want to know about the cell membrane. It's a phospholipid bilayer, as are all membranes, right? And what they do is they control what gets in and out. And this just has the single cell membrane. So the cell membrane here is controlling what gets in and out of the cell, right? When we get to the organelles in eukaryotes, each membrane around each organelle will control what gets in and out of that organelle. Cell wall, right? What cell walls do is they give a rigid structure, and under certain conditions, water could rush into a cell and plump it up, and so it gives a rigid structure so that it doesn't pop, right? And with osmolarity when we talk about it. Ribosomes, these are organelles. They're not bound on or in a membrane in the bacteria, so they're free in the cytoplasm. And they are the ones that are useful for making protein using the DNA clone. And remember, the ones in prokaryotes, and bacteria in particular, are smaller ribosomes. To the outside, we have three things that are farther out than the cell wall. They're made by the cell membrane and excreted to the outside. They are the capsule. There are pili, which is the actual cell membrane itself, and the fimbriae, which I'll go over in a minute. And then remember, it has a single loop chromosome with DNA. And its DNA structure doesn't have these histones. It's a little bit different than eukaryotic DNA, which is more of a 130 thing. The other thing we'll learn about is they have extra loops of DNA called plasmids. But those things will be in our next little lecture. And then flagella are useful for making it move in aqueous membranes or aqueous solutions. Okay, so cell wall. They maintain the shape. They protect the cell physically. They can block things from going in. They're more like a paper towel, right? And so they're not the screening mechanism for whether something gets in and out of the cell. They're more a physical block of larger particles, right? Remember, this is cell membrane that's controlling what's getting in and out of the cell. And here's written out for you. Remember, bacterial cell walls are made up of peptidoglycan, so that's one key feature, and that's one of the things we're going to be looking at in the Gram stain. It stains for peptidoglycan. And what it is a sugar molecule. Going back to cell walls, we find cell walls in bacteria, in archaea, and in eukaryotes that have cell walls, right? Each of the groups use different amounts of different types of sugar and amino acid derivatives, right? And so we have a certain particular structure that's peptidoglycan. You don't need to know that structure. That would be a more OChem thing. In bacteria, they all have peptidoglycan. In archaea, they have other substances than peptidoglycan that make up the cell wall. In eukaryotes, we have a variety of substances, the most common being cellulose and chitin. This last feature is important when we look at how they relate to each other. So I told you if I were to draw, like I did with that diagram, the prokaryote structure, you can tell a bacteria from an archaea. But if you were to look at, instead of that line where I just drew cell wall, but I told you the cell wall is peptidoglycan, you would know it's a bacteria. And in archaea, they do not have peptidoglycan. They have a variety of substances, but not peptidoglycan. So that's a definitive thing we could use for recognizing bacteria. Okay, in lab, I'll be going over more the procedure and how the Gram stain works. But while we're talking about the cell wall, within the bacteria domain, right, it can be divided into two groups. Those that are Gram-positive. And what a Gram-positive cell looks like, it's here on the left-hand side, it's more typical of a typical eukaryote cell as well that has a cell wall. You have the membrane, and to the outside you have whatever structural component is making up the cell wall. This being bacteria, it would be peptidoglycan. If this were a plant cell, it would look the same at this level. You'd have the cell membrane, but to the outside you'd have a cell wall of cellulose. If it were a fungus, you'd have a cell membrane, and the outside would be a cell wall of chitin. So this is very typical across all three of the domains. And what we do is we stain it with a substance that stains for peptidoglycan, and it stains it purple. And so this has a very thick peptidoglycan layer, and that Gram is called Gram stain. And so the Gram-positive ones will stain purple. So these little dots here are the Gram-positive bacteria. But then there's another group within the bacterial domain that is unusual in that it has a very, very thin peptidoglycan layer, and to the outside it has an outer second membrane layer. And this is showing you a phospholipid layer, right? So again, here's a phospholipid layer. Here are two layers with phospholipids, right? And this thin peptidoglycan layer is sandwiched between the two. And what happens in the Gram stain is we stain it with the purple dye, right? And then we wash it off with alpha alcohol. And just like any stain, so imagine, it's kind of dark, imagine you have red wine and you stain it on a white cloth, right? Even if you rinse it off with water, it still stains there, right? It's stuck onto it. So here what's going to happen in this thick cell wall peptidoglycan, that Gram stain doesn't rinse off and it remains purple. In this very thin layer, right, it's thin enough, first of all, the Gram stain will get in here and look purple, but once we rinse it off, right, it will go away and be clear. And so if we were to look at the slide, all you'd see were purple dots and everything else would be clear. So for the Gram negative, we add a very slight pink dye, and that's what allows us to see it. And the pink dye isn't staining for anything in particular, right? It is staining peptidoglycan, that's why you can see it. But here, because the Gram-positive are already darker purple, if you add a light pink to the dark purple, they still look at being purple, right? And when we get to the staining process, there's a little bit more involved. You use iodine as a mordant to help it bind and things. But that's basically looking at our Gram structure of the bacteria. Notice in the diagram up here, they're just on the outside. You can see the bare wall out here, so that's the peptidoglycan. And then here with all the ripples, they're showing you that that's a phospholipid membrane to the outside This membrane has proteins in it, and as well as the phospholipids, and some of those proteins can actually make them toxic. So generally, just as a broad generalization, the gram-negative bacteria tend to be more toxic or disease-causing than the gram-positive bacteria. Okay, so I went over that. So the complex cell walls, less peptidoglycan, they're thin, outer membrane, more likely to be disease-causing. Okay, if we go back to our diagram, things that I didn't draw on the diagram, they have a couple layers to the outside. And so here in orange is the cell wall. To the outside, the thicker area of yellow, that will be the capsule. Sticking through it, these yellow things are the same color as the membrane on the inside, right? And they'll be pili, and then what they're not showing you here are fimbriae, which will be little protein extensions. So we have three things that come out from the cell wall. Okay, so the capsule is a gelatinous secretion. It does a couple things, right? It helps protect them, kind of like sunblock, from drying out. It's kind of like putting a little extra gel layer that holds onto the water, right? It allows them to stick together, and in lab we're going to see a live culture of Gleocapsa. The capsa of Gleocapsa is referring to that thick capsule layer, and that's why it shows that cyanobacteria to take a look at. So hopefully in lab we'll be able to see the thick capsule layer. And there'll be individual cells all stuck together, making small little colonies, 4, 5, 6 cells stuck together in the capsule, right? It protects the cells also from things in the environment. And unfortunately, there's some disease-causing organisms that the capsule prevents our macrophages from actually getting onto them and attacking them. So it actually can, having a thick capsule can actually make it more disease-causing, right? And it could just be a general slime layer if the gel is less organized into an actual round capsule. Pili are a little bit different. So the capsule is this kind of, think of it like a jello type substance, a protein that's secreted out from the cell membrane, diffuses through the cell wall, then it just reacts with the water and the environment around it, and it turns into this kind of jello to the outside of it. That's what the capsule is, a gelatinous layer, right? It's protein with water. Pili are different. Pili are little micro extensions of the cell membrane that actually poke out and fold out from the inside of the cell to the outside. They do a couple things, right? And so they can give it more contact with the liquid environment outside the cells. But when we get to our next lecture, we're talking about how the DNA and how they exchange DNA. They make a little tube in conjugation, so DNA can go from one cell directly to another bacteria. So they're a little connection tube that goes across. And then lastly, we have little hard appendages that stick out. So the capsule is kind of like a sticky layer to the outside. These are little barbs, right, that will stick out. And these are called fimbriae, and they do what you would think they do. They help it stick onto surfaces, right? And so they can help it stick on, like if someone were to cough or these bacteria get in the air, they can get into your lungs. Your lungs cells are lined with mucus, but these can actually get through that sticky, slimy mucus and hold on to the cell individually. And so they help it attach onto surfaces. And so here's showing you bacteria with a thick capsule layer to the outside. This one is actually allowing it to get to and stick onto the tonsils, so that would be a bacterial infection of the throat. Here's one. These are fimbriae, and those are rigid proteins that are sticking out. Think of them as little thorns, right? So that's what they're doing. Is they're like on the bottom of a leaf. They'll brush by and stick onto your skin or your socks or something like that. And then this is a phospholipid bilayer going between two bacteria. And it will allow either a plasmid, which we'll talk about next time, to replicate and move over here, right? Or it allows for some of the DNA of the chromosome to replicate and move over here and merge and mix and recombine with the DNA of a chromosome in the other cell. This one's showing you lots of these pili extensions sticking out. Okay, I mentioned there are no membrane-bound organelles, but I do want to point out in some very special groups we're going to be talking about, and in particular two that will give rise to organelles in eukaryotes, what will become mitochondria and what will become in photosynthetic eukaryotes, plants, the chloroplast. And if we look at living bacteria, not organelles in eukaryotes, but living bacteria, some of them doing some aerobic respiration, right? We'll actually have, if you look here, this is the outer cell membrane, and notice how it's connected, and it just folds in. And so, if I were to draw my cell here again, see if I can remember what colors I use. So, I'll do yellow for, that's not yellow. Yellow for the cell wall. And then inside that, we have the cell membrane. Right? But notice, if I take that cell membrane, erase a little bit of it there. Not erasing for me. But if that cell membrane were to fold on the inside, right, that would be a little loop. I want to erase that little part, so let me erase. There we go. Anyway, I think it's because it made that structure, it's not doing that. That would still be the same loop, right? And if we make it more complex, here's our cell wall. Here's our membrane. I'm going to make it a squiggle so it doesn't make that little thing. It's going to fold in and around. It's still one cell membrane. It hasn't pinched off to be separate organelles. And so here, I just want to point out that we do see in the diversity of living bacteria some with very specialized membranes. So now what's happening is some of the membranes here are doing things different from the membrane out here. They have special structures, this is called thylakoids, that are different from the membrane out there. So they're not separate membrane-bound organelles yet, right? Because they still attach right here and right here. There are folds in the cell membrane. It's still one membrane, right? And it won't be a big jump. You can see with evolution how they pinch off and become organelles, but they're not quite there yet. So I just want to point out that even though they don't have membrane-bound organelles, they do have some special membrane on the inside. Both of these are examples of things when we talk about metabolism, and the way they do metabolism is using electron transport chains on the membrane. Okay, so they're formed by these folding in, what we call invaginations of the plasma membrane. Plasma membrane is the same as cell membrane, and they're not separate membrane-bound organelles yet. Okay, last one I want to talk about for structure is flagella. And what flagella do is they help single cells swim in an aqueous solution, right? And here are some bacteria showing you the flagella. They can be at either end. And we'll talk about next time how they move with the flagella and the motility. But one thing I want to point out, and if you remember from last time, I gave you two terms when we're talking about evolution. That was homologous and analogous structures. So take a minute and think if you can remember what the difference between homologous and analogous. So analogous, like an analogy, right? They're doing the same thing, right? And so the flagella of a prokaryote cell and the flagella of a eukaryote cell are analogous. They do the same thing. They have completely different structures, and they're not derived from a common ancestor, meaning the flagella of a prokaryote didn't turn into the descendant cell, the eukaryote's flagella, right? And they're very different structures, right? So a bacteria and archaeoprokaryote, they have a solid flagellum, right? It's not flexible, doesn't wiggle like a snake, right? It rotates at the base. It's more like a corkscrew propeller, rotating at the base with a little motor. Right? It's a very complex structure made up of several different proteins that had different functions in their origins, but came together over evolutionary time with modifications and became what is now the flagella. And I want to mention it's analogous because when we get to the eukaryote flagella, it has a completely different structure, and the way it works is completely different mechanism. Okay, so our next lecture will be going over motility. So we'll start with the flagella and talk about how they propel the cells in different ways and how they're attracted and not attracted. And then our third will be going over metabolism. Okay, and so please review this part of the chapter before we move on to the next one.